Hi everyone, my name's Suhani, and I'm here today to talk to you about the process of blood and its collection. Let's get started.
Since ancient times, humans have known that blood is vital to health. Blood transfusions have been occurring for centuries now. In 1900, Carl Landsteiner identified three human blood groups A, B, and O. In the years since, research on blood has been advancing rapidly, allowing us to identify even more blood groups. We also know more about the composition of blood. Whole blood contains red cells, white cells, and platelets suspended in blood plasma. White blood cells are one of the body's defenses against disease. They are collected by an automated process called apheresis and must be transfused into the patient within 24 hours of being donated. Platelets are small, colorless cell fragments in our blood whose main function is to stick to the lining of blood vessels to help stop or prevent bleeding. Red blood cells give blood its distinct color. They carry oxygen to our cells. Finally, plasma is the liquid portion of blood, and what our red and white blood cells are suspended in. It helps us maintain a satisfactory blood pressure and volume, and supplies critical proteins for blood clotting and immunity. So I'm here today with Nathan, who's a licensed phlebotomist, and he can explain this machine.
So this is a double reds machine, basically meaning, so what this machine does is it'll take out the donor's whole blood and it'll separate their platelets and plasma and red cells. So by the end of it, we just have a double red unit. So with this line, the needle goes in the donor and it gets separated. Go through these lines and it'll go in a little thing called a cassette. Once it's in the cassette, it'll know which one of these three tubes to go into. So this bag will have your whole blood in it, and then this will get siphoned back through this cassette into the centrifuge, and it'll get spun so fast that it separates the red cells with the platelets and plasma. And once that's done, it'll get sent back into this pack, which is just for platelets and plasma, and then with the red cells into this. And then every so often, it'll give a return to the donor. So whatever plasma is left, it'll go back through this cassette, back through this line, through the needle, back to the donor with a mix of citrate and citrate coagulant. And then once we have 360 milliliters of red cells, it'll start coming through this bag, through this cassette again, through this filter, into these two bags, which will eventually be our products.
Okay, perfect, thank you. Here are images of other cool equipment I saw today. All of these are used to separate blood into its individual components. The first is an apheresis chamber, which was mentioned before for white blood cells. The second shows a machine used to collect platelets. The final image shows the machine used to monitor the collection of whole blood. 9 out of every 10 individuals will need blood or a blood product at some point in their life. If you are able, check out your local blood bank today to see if you are eligible to donate. You could make the difference between someone's life and death.